one kind of lipid, there are all these little blue circles with the 2 feet hanging off, 2 legs hanging off. That's not really the case. It's more of a patchwork. It's not a uniform sea that's all the same. Lipids cluster. So for example, here is an atomic force micrograph picture of a cell surface. It's not just a uniform sea of the same thing. There's clearly different patches. These structures are called rafts. These are made out of lipids. So this is lipid and that's lipid, but they're different kinds of lipids. These rafts are made out of the sphingolipids. Remember, we had 2 classes of membrane lipids. Ones that are based on glycerophosphate backbone, and the other class based on sphingosine. Well, the sphingolipids and cholesterol like to cluster together to form these rafts.
Is that always the case? Is that, I mean, are those
It's concentration dependent,
but I mean, are those the only ones that form rafts? Or
no. Well, they call these rafts, but there's other kinds of clustering too.
Okay.
Here's an example of clustering not involving them. This is phosphatidylserine, the green ones. And they cluster when you add certain bivalent metals like calcium ions, causes clustering of those. The functional significance of that is not clear, but it's something that people have started noticing.
So when you say phase separation, you just mean?
Well, that's,
Yeah.
that may be a bad choice of words, you're right. But separating by type.
Okay.
So cholesterol is not uniformly distributed. In other words, sphingosine is not uniformly distributed. Proteins cluster too. Sometimes they have to do their job. And we'll look at some examples of that. Then there's transverse asymmetry. That's looking at outer leaflet versus inner leaflet. The proteins on the outside aren't the same as the ones on the inside. And even the lipids on the outer leaflet are different from the lipids on the inner leaflet. That's more of a surprise. It's certainly a surprise to me. So we show these as being the same, but it's not true. Here's some data on different kinds of lipids and their relative percentages on the inner monolayer versus the outer monolayer. Phosphatidylethanolamine, phosphatidylcholine have kind of opposite distributions. Why is there more phosphatidylcholine on the outside? It's a good question. People are looking at that, but it's been found now that that's true. Sphingomyelin, at least in this cell, is almost always on the outside. Why is that? There must be some reason for that.
So it could be related to a function of what needs to be done by the cell, right?
That's right. So there's an interesting story there that they haven't figured out. What is that function? What is it doing on the outside? We know a little bit about those kinds of stories for this one, phosphatidylserine. For many cells is often on the inside. What's this, maybe 90%, 95% on the inside, 5% on the outside. One curious thing that's been noticed with this is when phosphatidylserine starts moving to the outer part of the cell, the outer leaflet, that can trigger a process called apoptosis, or programmed cell death. So it's a cell death signal to move this lipid from the inside to the outside. So while we don't understand all of this, clearly there is some significance to the asymmetric distribution. Not to mention the fact that people have identified these so-called flipases and flopases. These are proteins that exist in the membrane whose sole job is to move a lipid from one side to the other, to flip it from inner to outer or vice versa, to maintain these differences. So the cell works hard to maintain this asymmetry. It spends energy doing it. Here's just some data. So there's not just one kind of asymmetry. It depends on the cell type. So here we have different mammalian cell types. So here's human red blood cell, liver cell, and platelet cell. Looking at different categories of lipids. This is phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine. These are sphingomyelins. So in the red blood cell, the sphingomyelins are almost all on the outside. If you jump over to the liver cell, it's flipped. It's almost all on the inside. So that's kind of weird. What does that mean?